Welcome to the Wednesday afternoon lecture series. I'm Ella Serpe, a senior investigator leading the section on cellular communication in NICHD. On behalf of the Women Scientist Advisors, I'm thrilled to introduce today's world speaker, Dr. Jessica Treisman, who has revealed incredible insights into cellular signaling through studies using the Drosophila model system. Dr. Treisman is a professor in the Department of Cell Biology at the New York University Grossman School of Medicine. She is a revered scientist in the cell biology and neurobiology community because she uncovered general fundamental mechanisms by which cells communicate. Using the visual system of the fruit fly as primary model, her group has identified novel components of several signaling pathways, including hedgehog, Wnt, Notch, and epidermal growth factor. She worked out the role of these new components during patterning and synapse formation and connected their misregulation to human diseases. Her group also studied how various signals are translated into cell phase decisions through transcriptional or post-transcriptional modulation of gene expression. From synaptic partner selection to contractile forces that control tissue morphogenesis to cell fate determination and synapse assembly, her insights are truly inspiring. Dr. Treisman received her undergrad degree from Oxford University and her PhD from Rockefeller University, working with Claude Desplan. After a postdoctoral fellowship in the laboratory of Gerald Rubin at UC Berkeley, Dr. Treisman moved back to New York in 1996 to take up a position as a faculty at the NYU Skirball Institute of Biomolecular Medicine. She quickly rose through the ranks at the Skirball Institute, becoming a full professor there in 2007 and director in 2020. In addition to her insightful research, Dr. Treisman is a stellar scientific citizen. She has served on numerous NIH study sections, has been an ad hoc member of the NICHD site visit review committees, has been a member of several editorial boards, and organized many important Drosophila conferences. And I want to leave you today with something I found fascinating and inspiring. Jessica's mother, Dr. Anne-Marie Treisman, was a famous psychologist who specialized in cognitive psychology. She gave a Walls talk in January 1997. As far as I and the Walls office can tell, today marks the first time a daughter of a past Walls speaker has followed in her mother's footsteps. Jessica, thank you so much for coming. We look forward to your talk. Thank you.
Well, thank you very much for that really kind introduction, and I am especially honored to be following in my mother's footsteps here. So today I'm going to try to tell you about a quite new project in my lab, in which we're trying to understand the development of the Drosophila corneal lens and to see if it can tell us something about the human cornea. And usually I don't have to spend too much time convincing an audience of scientists that the cornea is important, because I would bet that if most of you took off your corrective lenses, you would see this image looking something like this. And that's because the cornea contributes 70% of the refractive power of the eye, and so defects in the cornea lead to many different kinds of visual refractive errors. So if the cornea is too strongly curved, it focuses the image in front of the retina, giving rise to myopia. If it's not curved enough, the image will focus behind the retina, giving rise to hyperopia. And if the curvature is uneven, then the image will be distorted, and this is astigmatism. So the Fly Corneal lens at first glance might look rather different from the human cornea, but they do have some things in common, and one is that they're mostly composed of extracellular matrix. So if you look at a section through the human cornea, you can see that about 90% of the thickness is this stromal layer, which is an extracellular matrix made up of collagen fibers and small leucine-rich proteoglycans with interspersed keratocytes and cellular layers above and below it. And the Fly Corneal lens is actually completely acellular extracellular matrix. In this case, the fibers are made up of chitin, which is a polysaccharide, and their associated cuticle proteins. But we really know very little about the composition of this corneal lens and why it develops its precise curvature. And we've begun to tackle this, and one of the first steps that we took was to try to understand how the cells that secrete this corneal lens develop. So these would be the underlying cone cells and pigment cells. So this is where there is a significant difference between the Drosophila and mammalian systems. So the mammalian cornea develops from the surface epithelium after the lens has invaginated, and it's quite separate from the retina, which develops from the neural tube. In flies, a single epithelium, the imaginal disc, so the imaginal disc gives rise first to the 8 photoreceptors in each unit eye of the compound eye. So these differentiate in the order R8, R25, R3 and 4, R1 and 6, and then R7. And then the same epithelium will give rise to the 4 cone cells and 2 primary pigment cells and a lattice of secondary and tertiary pigment cells which secrete the corneal lens. And so this happens as a wave of development progresses across the imaginal disc, giving rise to all of these cell types in succession. And this means that an undifferentiated progenitor in the anterior of this eye disc can give rise to a number of different cell fates. And this is a question my former student Carolyn Morrison was interested in understanding. How is it that a single progenitor could give rise to either a photoreceptor, which is a light-sensitive neuron, or a cone cell, which is a glial cell that can secrete corneal lens components, or a pigment cell, which will produce screening pigments and recycle retinal in the visual cycle. So Carolyn had one clue to start with, which was a protein that was discovered back in 1989 by Kevin Moses in the Rubin lab. And this is a protein called GLAS. It's a zinc finger transcription factor. And you can see that in a GLAS mutant, the eye is much smaller and more disordered than a wild-type eye. And if you section through an eye that's a mosaic of both wild-type and GLAS mutant tissue, you can see that in the wild-type region, you see the photoreceptor rhabdomeres. So these blobs are the light-detecting organelle that's analogous to a rod or cone outer segment. But in the GLAS mutant region, these rhabdomeres are completely missing. And so it was thought that GLAS specifies the photoreceptor sulfate. And indeed, if you look at photoreceptor-specific genes, they seem to be completely absent in GLAS mutants. So for instance, chaoptin is expressed in photoreceptors in the larval imaginal disc and missing in GLAS mutants. Rhodopsin-1 in the adult retina is also completely gone. So these are a couple of genes, but we wanted to get a more complete picture of what GLAS was affecting. And so we collaborated with Graham Marden at Baylor and Kelvin Young and Raja Kumar in his lab. And they did a single-cell RNASeq analysis of wild-type and GLAS mutant discs. This was at the end of the larval stage. So in the wild-type eye discs, they could see separate clusters representing each of the photoreceptor cell types. So R8, R2 and 5, 3 and 4, 1 and 6, and R7, as well as the cone cells. And each of these clusters is actually a trajectory from less differentiated on the left to more differentiated on the right. In GLAS mutant discs, on the other hand, the only real trajectory that was still visible was the R8 trajectory and a smaller cone trajectory. So there are early markers of R2, 5, 3, and 4 photoreceptors, but no extended trajectory of differentiation. And R1, 6, and 7 seem to be completely missing. So I'll just show you a few of the genes that conclusion is based on. So senseless is an R8-specific gene. That's how we know that this particular cluster represents R8 in both the wild-type and GLAS mutant. And if we look at early photoreceptor-specific genes, such as scratch, you can see that this is coming on in all the photoreceptor trajectories in wild type. And in scratch, we see it in R8, and it also turns on in this region where R2, 5, 3, and 4 are just beginning to differentiate. But later photoreceptor-specific genes, such as retained, in the GLAS mutant are only present in R8. So GLAS seems to be necessary for the differentiation of all of the photoreceptors other than R8. So that's what GLAS is necessary for. Carolyn wanted to see what it was sufficient to do. And so she misexpressed GLAS in a different tissue, the wing disc, to see what it could induce in that ectopic situation. So here, the green cells are expressing both GLAS and GFP in a wing imaginal disc. And she saw that K-optin, that photoreceptor-specific gene, was turned on by GLAS. In a wild-type eye disc, K-optin in red here would only come on after the cells had already been expressing neuronal markers, such as LV, for quite a while. But in the wing disc, LV was not induced by ectopic GLAS. So GLAS can induce photoreceptor-specific genes, but it's not really transforming the cells into photoreceptors because they're not taking on a neuronal identity. And this isn't completely surprising because GLAS expression in the eye disc is not restricted to the photoreceptor neurons. It's actually expressed in all of the cells in the posterior differentiating region of the disc, as you can see here. And so GLAS can't be sufficient to induce the photoreceptor neuron identity. There must be something else working together with it. And we had a good candidate for that, which is the Epidermal Growth Factor Receptor signaling pathway, which Matthew Freeman and Kevin Moses, among others, had shown was essential for photoreceptors other than R8 to differentiate. So as this differentiation wave moves across the eye disc, hedgehog signaling turns on atonal, which is a proneural factor, and that specifies the R8 photoreceptor. And R8 then produces Spitz, which is a ligand for the EGF receptor, and it's EGF receptor signaling that then induces R1 through R7 to differentiate. So if you knock out EGFR signaling, you lose the photoreceptors other than R8, so similar to the GLAS mutant phenotype. So this is just a simplified version of the EGF receptor signaling pathway, and I just want to draw your attention to the transcription factor downstream of the pathway, which is called pointed. And an early step in the pathway, the GDP binding protein RAS, which we can use to activate the pathway because there's a constitutively active form of it. So we looked at whether this pathway could synergize with GLAS to induce neuronal differentiation in the wing disc again. So GLAS alone does not turn on our neuronal marker LV. Activated RAS alone turns on LV in a few cells, but not significantly. But if we express both together, now a lot of cells take on this neuronal identity, expressing LV. And we could show that this is dependent on that downstream transcription factor pointed, because if we misexpress GLAS and activated RAS in cells that are mutant for pointed, we lose most of that ectopic LV expression. So it seems to be a synergy between the two transcription factors, GLAS and pointed. So LV is just one gene, so maybe I shouldn't be making such a broad conclusion based on that. My student, Hong-Soo Wang, wanted to know what else GLAS and activated RAS could induce, and so she did RNA-seq on those wing discs that I just showed you to look at what genes were induced by the synergistic interaction of GLAS and RAS, and she found 256 genes that were synergistically induced by GLAS and activated RAS more than either alone, and that were pointed dependent. And at least 40% of those genes have known neuronal functions. So this is just a few examples. Nerfin1 and scratched neuron-specific transcription factors synaptotagmin 4, a neuronal synaptobrevin, a synaptic vesicle proteins. TRIP is a channel. It's important for phototransduction. And FRITch is a microtubule-associated protein specific to neurons. So all of these are induced much more strongly by the combination of GLAS and activated RAS than by either alone. And the expression is much reduced in pointed mutant cells. So next, Hong-Soo wanted to know how this synergy was actually happening in a mechanistic way. And there are two sort of extreme possibilities for that. So either there could be a direct synergy, which both GLAS and pointed would be directly binding to the promoters of all of these downstream genes. Or alternatively, either GLAS or pointed or both could act primarily by inducing other transcription factors, and those transcription factors would then act directly on these downstream target genes. So to address this question, she used a technique called targeted DAM-ID. This is a way to look at transcription factor binding sites in a cell-specific way. So the idea is that you fuse the transcription factor that you're interested in to the DAM methylase from E. coli, and that can then methylate the DNA where that protein is bound. And you can express this in specific cell types at a very low level so that it doesn't disrupt cell fate or anything. And you can use methylation-sensitive restriction enzymes to then isolate and sequence that DNA. So Hong-Soo did this with both GLAS and pointed, so she fused each of those to the DAM methylase, as well as expressing DAM methylase alone as a control, just to show where open chromatin is. And she did this in 3 different cell types. So atonal GAL4 is expressed in the first cells to differentiate in the eye disc. LV GAL4 is specific for the photoreceptors, and sparkling GAL4 is expressed in the cone cells. So the idea was to see what genes GLAS and pointed can bind to in all of these different cell types. So we just actually got these data a couple of weeks ago, and so I can't really tell you the full analysis yet. But I just wanted to show a few preliminary conclusions. So first, we can say that GLAS and pointed have both shared and distinct targets. So for instance, both GLAS and pointed bind to the lozenge gene. And interestingly, this second intron was already shown to contain an enhancer that's necessary and sufficient for lozenge expression in the eye disc, so that was nice. Some genes like prune are only bound by GLAS and not pointed, and other genes like Dusky-like are bound by pointed but not GLAS. Also, if we look at the different cell types, there are some genes that are regulated or bound by GLAS in all cell types, the early differentiating cells, the photoreceptors, and the cone cells. And others are only in the early cells, or only in the neurons, or only in the cone cells. And similarly for pointed, there are some genes that are bound by pointed in all 3 cell types, or only in early cells, neurons, or cone cells. And to come back to those synergistically induced genes, there are some that seem to be directly regulated by GLAS and pointed binding to the same site, such as FRITch. You can see binding by both GLAS and pointed. And there are others that appear to be indirectly regulated because we don't see binding of either GLAS or pointed. So there must be a secondary transcription factor that's mediating the effect of GLAS and activated RAS on the expression of NERFIN1. Okay, so to come back to this point that I made earlier, that GLAS is not restricted to the photoreceptors, it's also expressed in the other cell types in the eye disc. We wondered whether it might have some function in those other cell types as well. And we could show that it does seem to. So if we look at the genes that are induced by GLAS when it's ectopically expressed in the wing disc, we can find genes that are specific for cone cells, or primary pigment cells, or secondary and tertiary pigment cells. And these are all induced by GLAS independently of RAS activation. So RAS activation does not further induce their expression. And we also knew that glass has biological effects on these non-neuronal cell types. So if we knock out glass autonomously in the pigment cells, we lose the red eye pigment. And if we misexpress glass in the leg or other sites on the body, we can see ectopic induction of red eye pigment. So glass seems to be specific to induce at least some aspects of pigment cell identity. Antonia Terrazano, a master's student, wanted to know if glass was really directly regulating these genes that are specific for the non-neuronal cell types. And so she tried to clone out some enhancers upstream of genes that we knew were glass-dependent and had predicted glass binding sites. And she was able to find some that were specifically expressed, or it specifically drove reporter expression in cone cells, and others that specifically drove reporter expression in pigment cells. So here we see the primary pigment cells and the secondary and tertiary pigment cells. And this expression is glass-dependent because if we make glass mutant clones in these retinas and the glass mutant regions are marked with GFP, we see loss of reporter expression driven by these enhancers. So we still need to mutate these glass sites in order to show definitively that glass is directly regulating these enhancers. But we think that they'll be useful to help us understand why glass would turn on different genes in different cell types. So why does it turn on this one only in cone cells, this one only in pigment cells, and other genes only in photoreceptors? And we imagine that there are other transcription factors that bind to these enhancers and account for these differences. And we do know a few of those transcription factors just based on our guesses. So for instance, escargot is a pigment cell-specific transcription factor, and it seems to synergize with glass to turn on pigment cell genes. So if we misexpress glass, it can induce red pigment on the abdomen. But if we misexpress glass in cells that are mutant for escargot, it's not able to induce that pigment. And ectopic glass in the wing induces only a little bit of pigment, but if we express it together with escargot, we see much more pigment induction. So we think that glass and escargot together may specify pigment cell genes. And we've also identified some other transcription factors that may control photoreceptor-specific or cone cell-specific genes together with glass. So that's where we are at the moment with the cell fate question. And now I'd like to turn to the question of how the corneal lens itself is actually produced by these cells. So I realize that this is quite an understudied area. We actually know very little about this corneal lens. It is a cuticular structure, and so we can get some clues about how it might be composed by looking at other cuticles, like the larval cuticle, the body covering. And that cuticle is made up of 3 cell layers. So there's an outer envelope that has waxes and proteins. It's a protective layer. There's an inner procuticle that contains the chitin fibers. And in between, there's an epicutical layer marked by Tweedle proteins, among others. Now, if we look at some of these markers in the corneal lens, chitin seems to be strongly enriched on the inner surface of the lens. So that's where we think that the corresponding layer to the procuticle might be found. But when we marked an endogenous Tweedle protein, hoping to label the epicuticle, we found that it was actually localized to the periphery of these corneal lenses and not in a central layer as we thought it might be. So it's really not clear whether the corneal lens has the same composition as the larval cuticle or what the bulk of this lens is actually composed of. So if we don't know that, it's hard to tell how it develops its precisely curved shape. And our first handle on this came when Hong Su was investigating another transcription factor, a zinc finger transcription factor called Blimp1, which is actually quite related to glass. And I should say Gerald Cole at Midwestern University and his student Joy T developed some of these results at the same time, and we collaborated with them. So when they knocked out Blimp1 by making a mutation using CRISPR, they found that Blimp1 mutant clones in the adult eye had a rather smooth and glossy surface where you couldn't really distinguish individual corneal lenses as you can in a wild-type eye. And if you cut sections through this eye, you can see that in the wild-type regions, the lens is curved on both sides, so it's biconvex. But in the Blimp1 mutant region, which is marked in red here, the external surface is completely flat. So this is a plano-convex lens. And Hongsu calculated the change in focal length that this shape change would cause and found that the focal length is very significantly increased, so much so that these lenses would not be able to focus light within the whole thickness of the retina. So a fly that had completely Blimp1 mutant eyes would probably not be able to see with much precision. So we looked at where Blimp1 is expressed, and we found that it's most highly expressed in the non-neuronal cell types in the retina. The cone cells, primary pigment cells, and the secondary and tertiary pigment cells. And it has a peak of expression about halfway through the pupil stage. This is the time at which there's a big peak of ecdysone. Ecdysone is a steroid hormone that controls developmental timing in Drosophila. And we could show that Blimp1 is actually dependent on ecdysone. So if we express ecdysone receptor RNAi to knock down ecdysone signaling in clones in the retina marked with GFP, we saw a loss of Blimp1 expression. Okay, so it's a developmental timing signal in these cells. So we thought that Blimp1 would be required in the cells that are known to secrete major components of the corneal lens. So the cone cells and primary pigment cells are directly underneath the center of the lens, and they are known to secrete crystalline and other major corneal lens components. But actually, if we knock out Blimp1 expression specifically in those cells by expressing Blimp1 RNAi with the driver that's specific for cone and primary pigment cells in this RFP-labeled region, there is no major change in the shape of the lens. So we found that the cells that actually do require Blimp1 to control the corneal lens shape are these secondary and tertiary pigment cells, which are found at the periphery of the corneal lens. So if we knock down Blimp1 specifically in those cells, now we do see corneal lenses with a flat external surface. And you can see this more clearly if we do it in the whole eye. So the outer surface of the lens is really completely flat. So we were wondering, how is it that these cells that are right at the edge of the lens could have such an influence on its shape? And one idea we had was that these cells are actually attached to the periphery of the lens, and maybe they could be exerting some kind of mechanical force on the lens that would change its shape. And if we zoom in on this region where the pigment cell attaches to the corneal lenses and look by electron microscopy, we could see that there does seem to be a change in the number of points at which the pigment cells are attached to the corneal lens. So there seem to be more of these attachment sites when we knock down Blimp1 in the pigment cells. So that makes it plausible that there could be some change in the forces that these cells apply to the corneal lens. But in order to get a mechanism, we needed to know what genes Blimp1 is regulating in these secondary and tertiary pigment cells. And so Hongsu again did an RNA-Seq analysis of retinas in which Blimp1 was either knocked down or overexpressed. And she found many different types of genes that were regulated by Blimp1. But one class of genes that caught our eye was a set of genes that encode Zona Pellucida domain proteins. So some of these are upregulated when Blimp1 is knocked down, and others are downregulated. So these proteins were interesting because they were known to control the shape of other cuticular structures, such as the denticles that are found on the ventral side of the embryo. So Francois Per's lab had shown that a number of different CP domain proteins are expressed in these denticle-producing cells, and each one of them has a different effect on the shape of the denticle when you remove it. So Neha Ghosh, a postdoc in the lab, started looking at whether any of these CP domain proteins had phenotypes affecting the shape of the corneal lens. And the most striking one she's found so far is for a gene called Dusky-like, or DYL. And this is a protein that's found at the very tip of those embryonic denticles. So each protein is localized to a different region of this apical extracellular matrix. So she found that when she made DYL mutant clones in the retina, marked by GFP here, the corneal lenses again changed their shape. So the external surface is flatter, and the internal surface is more deeply curved. And we can quantify this by measuring the outer angle between two adjacent corneal lenses. So in both Blimp1 and DYL mutant regions, that angle goes up to almost 180 degrees, so almost completely flat. And the DYL mutant phenotype can be rescued by re-expressing DYL in the mutant cells. And I didn't show it here, but she's also measured the inner angle and showed that it's decreased in the DYL mutant clones. So to understand how this change in shape was happening, Neha went back to earlier stages of development. So she looked at the mid-pupil retina. And at this stage, these cells form a beautiful repeating pattern at the apical surface of the retina. So you see 4 cone cells in the middle, surrounded by 2 primary pigment cells, and then secondary pigment cells on each edge, and tertiary pigment cells or bristles at the corners. So when she made DYL mutant clones, which are marked with GFP here, and DYL protein is marked in red, you can see that DYL protein is localized to the apical surfaces of these cells. And in the DYL mutant regions, those apical surfaces are smaller. So you can see, for instance, here, that this mutant primary pigment cell is smaller than the wild-type one on the other side of the ommatidium. And the same thing here and in other places. And the completely DYL mutant ommatidia are smaller overall. And a few hours later, this effect becomes much more pronounced. So the DYL mutant green ommatidia are significantly smaller than wild-type ommatidia in the same retina, and are also no longer in the same apical-basal plane. And that's because apical constriction usually goes along with apical-basal contraction of the cells. So if we cut a cross-section through the retina at a slightly later stage, you can actually see this apical-basal shortening of these GFP-labeled DYL mutant cells more clearly. So they're becoming shorter in the apical-basal dimension than the nearby wild-type cells. You can also see an accumulation of actin at the apical surface. So this is labeled with myosin, an actin-binding protein. And you can also see that there's a change in the shape of the developing corneal lens, which is bulging more inward than the wild-type lenses, which are bulging outward. So we think that this shortening of the ommatidia may actually pull on the corneal lens to mechanically change its shape. So our model based on this is that DYL at the apical surface of these cone and primary pigment cells may be attaching it to the developing corneal lens, and that these apical surfaces would be expanded and kind of under tension, so that if you get rid of DYL, those cells would shrink the apical surfaces, they detach from the lens and kind of contract downwards. And we think that the secondary and tertiary pigment cells are still attached to the corneal lens, and so they would pull the corneal lens inward as these ommatidia contract. So we wanted to try and test this model, and to do that, Neha induced apical constriction in an artificial way that had nothing to do with DYL. So she misexpressed an activated form of myosin light chain kinase in clones of cells marked with GFP. And you can see the same kind of apical constriction in these clones, so that the apical surfaces of the ommatidia that are expressing activated myosin light chain kinase are smaller than nearby wild-type ommatidia. And when she looked at the adults that developed, she saw that, again, in these mutant regions, the shape of the corneal lens was changed. So again, it was bulging inward much more than the nearby wild-type regions. So apical constriction in itself is sufficient to change the shape of the lens and increase the inner curvature. Okay, so however, there's still a problem with our model, and that is that DYL is not there for very long. So it has this very sharp peak of mRNA expression, according to Flybase, just at this mid-pupil stage. And we can see the same thing if you look at the protein. So its highest level of expression is at 50 hours of pupil development, and just a couple of hours later, there's much less protein, and then it disappears. So DYL itself can't really be forming a very long-lasting attachment of the corneal lens to the underlying cells. So we know that ZP domain proteins can polymerize into long filaments. So this is showing the structure of uromodulin, which is a ZP protein, and an EM structure of the filaments that it forms. And we also know that different ZP proteins can form filaments together. And so we thought that perhaps DYL might be affecting the organization of other ZP domain proteins. So we looked at a few of them. So one that was known to form filaments is Dumpy. This is a really enormous protein that has a ZP domain. And Shigeo Hayashi has shown that at muscle attachment sites, it forms very nice filaments that connect the muscle to the tendon. So Neha found that if she looked at Dumpy in the developing corneal lens, it also seemed to be forming some kind of filamentous structure. And this organization was dependent on DYL. So in DYL mutant regions, we don't see these filaments forming. And she saw a similar thing with two other ZP domain proteins. So POPO, again, forms filaments in wild-type regions, but not in DYL mutant regions. And the same thing for Trinity, which is another ZP domain protein. So again, there seem to be filaments in the wild-type, but not in the DYL mutant regions of the retina. So this may be part of the answer to what DYL is doing, but it's likely more complicated than that, because if we look at another non-ZP domain component of the corneal lens, so the Tweedle E protein that I mentioned before that's found at the periphery of the lens, that doesn't seem to be secreted or deposited properly in DYL mutant clones. So you can see it in the wild-type regions, but in the DYL mutant region, more of it seems to be stuck inside the cells. So we think that DYL is helping to assemble a network of ZP domain protein filaments, including Dumpy, POPO, and Trinity. But it also may be forming some kind of conduit for the secretion or deposition of other corneal lens components, such as Tweedle E. Okay, that's all well and good, but we haven't completely solved the problem of corneal lens shape yet, because even though DYL is regulated by Blimp1, it doesn't completely account for the effect of Blimp1 on corneal lens shape. Because if we look at Blimp1 mutant clones at the same stage, this mid-pupil stage, the Blimp1 mutant cells, which are marked in red here, actually expand the apical profiles. So they're bigger than neighboring wild-type cells. And also, rather than just changing the organization of dumpy, they don't seem to secrete much dumpy at all. So Blimp1 is certainly doing other things than just regulating DYL. So one class of Blimp1 target genes that we were interested in looking at were those that are repressed by Blimp1. So Blimp1 is known to act primarily as a repressor in some other systems. So in Blimp1 mutant cells, these repressed targets would be upregulated. And so we reasoned that if we were to downregulate those misexpressed proteins, we might be able to rescue the phenotype of Blimp1. And this was probably too simplistic because there's so many targets of Blimp1. It was perhaps unlikely that we would find a single one that could rescue the phenotype. But we actually did find one that had a pretty strong effect. So this is a protein called CG4333. We have to give it a better name soon, but it was upregulated in Blimp1 mutant or Blimp1 knockdown retinas. And it seems to rescue the phenotype. So again, Blimp1 mutant clones have externally flat corneal lenses. But if we make Blimp1 mutant clones in which we knock down CG4333, they have a much more normal curvature. From the outside, the eye doesn't have those smooth, glossy patches. And if we measure that outer lens angle, it's restored to a more normal angle. And this was especially interesting because CG4333 is homologous to a human gene called TGF beta-induced protein, or TGFBI. And mutations in TGFBI give rise to many human corneal dystrophies. So TGFBI and CG4333 are both fasciclin domain proteins. And within those fasciclin domains, there are many amino acids that are both conserved between these two proteins and can mutate to give human corneal dystrophy. And Shahrukh Khan, a technician in the lab, is now working to knock in some of these point mutations that give corneal dystrophy into CG4333 to see if we can get an eye phenotype. But another thing that we do know is that many of these TGFBI mutations that give rise to corneal dystrophy have that effect because they cause accumulation or aggregation of the protein. So they can either affect its solubility or its turnover by ubiquitin-mediated degradation, or they just induce aggregation of the protein. And that causes accumulation of TGFBI protein and corneal dystrophy. And so we wanted to see whether just overexpressing CG4333 might have an effect. And in fact, it does appear to flatten the external surface of the lens. So we're hopeful that there might be more homology between the corneal lens and the human cornea than might appear at first glance. And that, thinking about conservation, made us go back and take a look at Blimp1, because the Jester lab had shown that Blimp1 is expressed in the human cornea. Well, actually, this is mouse cornea, sorry. But it's specifically localized to the limbus. And the limbus is the region where the curvature of the cornea initiates. It's also where adult epithelial stem cells are found. So we were interested in learning more about the function of Blimp1 in the cornea. And so we started a collaboration with Shukti Chakravarti at NYU. And she's interested in diseases of corneal curvature, such as keratoconus, in which the cornea is too curved. And she uses both mice and human corneal organoids in order to study this. So we thought this would be a great system in which we could look at the function of Blimp1. So we haven't got very far with this, but she has found that from her single-cell RNASeq analysis of both human cornea and human corneal organoids, that Blimp1 is expressed in a subset of the epithelial cells in both the corneas and the organoids. And her postdoc, Sean Ashworth, has started to look at Blimp1 target genes, beginning with a human corneal epithelial cell line. And we're still waiting for the genome-wide analysis. But he has found at least one gene that's regulated by Blimp1 in both systems, which is perlecan. So So that's encoded by HSPG2 in the human cornea, and that's reduced when he knocks down Blimp1 with siRNA. And in flies, the gene is called TROL, and we also saw a reduction in TROL when we knocked down Blimp1 in the fly retina. So we're hoping that there are going to be more common targets that will help us understand whether we can use the corneal lens to model the human cornea. So, yeah, so I hope that, I mean, it's still early days, but we do hope that we can use this corneal lens, not only to understand how you can make a curved structure out of extracellular matrix, but also how the curvature of the human cornea is generated. And we're hoping to find some more common features. Perhaps it will turn out that TGFBI in the cornea is repressed by Blimp1. There might be roles for ZP domain proteins, perhaps. There's a ZP4 is expressed in the corneal endothelium. So it would be interesting if it turned out to also have a role in attachment of the cornea. And yeah, we're looking forward to investigating possibly conserved mechanisms in corneal development and disease. So with that, I'd like to thank my lab members. So I talked about the work of Neha Ghosh, Hongsu Wang, Antonia Terrazano, and Shahrukh Khan, and a former student, Carolyn Morrison, and our collaborators, Gerald Kaul, Shukti Chakrabarti, and Graham Marden. And of course, I'd really like to thank NIH for funding these projects. So thank you.
That was quite the story. I got the feeling that DIL is one of these Zona Pellucida proteins that must be tightly regulated. Too little and too much can create havoc in fiber formation of the other Zona Pellucida proteins. I was wondering, you showed us how DIL loss of function create more constriction. Could we assume that overexpression of it will relax?
So that's interesting. That is something that we should probably look at. So we haven't really done much analysis of DYL overexpression, but that would certainly be interesting to look at because it does seem to be extremely tightly regulated. So.
Thank you for a very nice and very clear talk. This isn't my area of expertise, but I was wondering, you talked a lot about how these proteins that shape the cornea are regulated during development. Do they change in the adult after development? Because obviously there seem to be changes in the corneal structure in humans anyhow after birth. And so are there changes in these proteins in the adult fly?
Yeah, that's another thing that we haven't really looked at very much yet. So I don't think anyone has much studied how the corneal lens might change with age, because it is just this completely acellular, extracellular matrix structure. And so I don't know how much turnover there is of the components and whether they keep being deposited in the adult or not. That is something that we've considered looking at with this CG433 mutations, because corneal dystrophy is something, it's definitely an age-related disorder that gets worse with age, and different mutations have different onsets in humans, so we are considering aging those flies once we make the mutations and seeing if age has any phenotype.
That was a fantastic talk. I had a couple questions about your glass binding results. One of them was, does the leg express the cofactor that's in the pigment cells for the pigment-specific genes? Is that why the trick of putting glass in the leg works to turn on all these pigment genes? And then the other one is, I think you had several glass targets that were shared across all cell types. Do you think that there's a ubiquitously expressed cofactor for those genes, or do you think that there's a difference in the nature of the binding site that lets glass bind on its own sometimes, but need a cofactor other times?
Those are great questions. So escargot is that pigment cell gene, pigment cell transcription factor I mentioned. So that is expressed in the leg. It's mostly in the most proximal leg, but it does very early on, it would be in a larger region of the leg disc, and so that may be why we don't see this pigment necessarily restricted to the most proximal part of the leg. And yeah, as for the genes that are regulated by GLAS in all cell types, we're definitely going to have to look more closely at what they are. We're just getting these data right now, and see whether. So I don't know whether other transcription factors would be necessary for GLAS to bind, though, or whether they would just be necessary for transcription to be activated. So binding doesn't necessarily mean that there's an effect on the transcription, so we're going to have to really correlate those with our data on what genes require GLAS for their expression to be able to figure that out.
I've been asked to repeat the CME code. It's 44453. And Dr. Sherpa, if you have any closing remarks.
Well, if there are no more questions, I would like to thank Jessica once more for a fantastic talk. And to invite all of you to stay around a little bit for more questions and coffee and cookies right outside the amphitheater. Thank you very much.
Thank you.